And I have to say, and Johannes said that in 1984 he began in terms of looking at RFLPs in Factor VIII, and at the beginning of my research career was in 1985 looking at RFLPs and von Willebrand Factor, so we kind of have a common start. So it's my pleasure today to talk to you. The title of my talk is Genotypes, Phenotypes, and Whole Genome Sequence, and I'm going to tell you a bit about the MLOF, our future project in the US, and then some initial results from our involvement as a cohort in the US TopMed program. And these are my disclosures. So what is MLOF, our future? So it's a US national program for the hemophilia community. We offered free genotyping to people with hemophilia and their families. There were 2 surveys, one to the patient community in the US and one to the provider community in 2012, and unfortunately only about 20% of our patients had been genotyped. And we also want the knowledge from this project to improve hemophilia care. And I'll also tell you more about a repository we have as part of this that can be used for research going forward. And the project is a collaborative project with the American Thrombosis and Hemostasis Network, Bloodworks Northwest, where I am from, the National Hemophilia Foundation in the US, and BioVeritiv, which funded the project. So the process, this took place at 111 hemophilia treatment centers in the US, and patients or parents were approached at the hemophilia treatment center and authorized testing, which was drawn and then sent to our laboratory at Bloodworks Northwest. At that time, the patients or parents were asked if they would be interested in participating in the research repository, which the consent included consent for whole genome sequencing and deposition of data into large databases. And so for those who gave written informed consent, they had additional samples collected and sent to us. And also important in this project is the American Thrombosis and Hemostasis Network, or ATHN, a database which collects phenotypic data on all of the patients and those who had with this given consent for their data to be part of a HIPAA-compliant data set. And thus we have the power to have genotypic and phenotypic data. So this is the enrollment. The enrollment in this phase of the project ended in December. So we had a gradual increase in number enrolled, but you'll see there's an inflection here and a sharper increase. And this is when we invited enrollment of women who were potential or known carriers, genetic carriers of hemophilia. So I think this also represents a continued unmet need in that population, as we didn't come close to leveling off. So total, we enrolled 11,356, and reflecting the interest in research in the community, over 9,000 gave informed consent for participation in the research repository. So the patient characteristics, now what I show you on the table here does not include data on the 1,468 confirmed genetic carriers in whom we have factor activity levels that are known to be normal or we don't know yet, and on the 633 females who were confirmed to be non-carriers. Now, the same percentage of those women agreed to the repository, which I think gives it further strength because we have unaffected females in there also. So we, both males and females with hemophilia defined as less than 50% tested at their local HTC were eligible. We have almost 6,000 males with hemophilia A and about 500 females. similar percentages in the repository, and it shows that we have a distribution of severities in the males. In the females, we have more milds, but we have some females with severe and moderate disease in both hemophilia A and hemophilia B. Our approach, we used a next generation sequencing approach using molecular inversion probes as our capture, which actually gave us the ability to do something with inversions, which was fun. We did this in collaboration with Jay Shinduri's lab at the University of Washington. And we sequenced Factor VIII and Factor IX at the same time, which is a small number of genes for this technology, but that was the focus of the project. And so we have these targets that capture the coding region and the immediate upstream, downstream region and the intron flanking areas. But we also used inverse shifting PCR as described by Rossetti and colleagues. We adapted that to this process. And so in this, there's a digestion and ligation, and we could target the ligation product of the intron 22 and intron 1 inversions also with MIPS. And so when we mixed them all together, we could detect Factor VIII, Factor IX sequence, and we could detect whether the inversion was present at one time. And so then they're all mixed together and they're sequenced, and then because this process at this time is not CLIA certified in the US for a laboratory for reporting variants, we confirmed the variants using a standard approach, and we also used MLPA for structural variants if no variant was found, or if we found only one variant in a female with a low factor level. And the variants that we found are those that have reported. The Factor VIII and Factor IX genes have been sequenced by many people and reported, and so there was not a surprise in the type of variants we found. In terms of the females with moderate and severe disease, a number of them, but certainly not all of them, had two likely causative variants. Now, usually we can't say for sure that they have a variant, whether the variants are on one allele or on both. But in 15 females with severe A, 4 had two likely causative variants, and moderate, similarly, we only had so far one female with severe B, and she is hemizygous due to Turner syndrome. And then we had 3 females with moderate B, and all of them we only found one causative variant. Overall, in all of our so far documented genetic carriers, in hemophilia A, 39 females with 2 likely causative variants, and in hemophilia B, 6 females with 2 likely causative variants. One of the surprising things we found, we have reported previously on our first 3,000 subjects, and because there's been so much genotyping in hemophilia, we thought we would find saturation at some point, but in the first 3,000 patients, we found 300 previously unreported variants, and a very straight line. And overall, to date, in just over 8,000 subjects, we have found 720 novel variants, so not quite at the initial rate, but very close. So there's clearly a lot of genetic variation that can be causative in the Factor VIII and Factor IX genes. So one of the things we've been able to do is to, because we're sequencing Factor VIII and Factor IX in all patients, we have found some variants in the other gene. So if the patient has hemophilia A, we find a variant in Factor VIII, and a few of these, we also find a variant in Factor IX. So some of those are known to be benign variants. And actually, the reverse is more common just because Factor VIII is a bigger gene than Factor IX. And so if we find a variant in the other gene that has been previously reported in hemophilia or appears to be likely or pathogenic. Obviously, then we communicate back to the HCC and ask for them to obtain a factor level of the other the factor activity on the patient if they've not previously done so. And to date, we've confirmed 12 variants previously reported as causative in the hemophilia databases to be benign variants. And so I think when we started genotyping in hemophilia, we thought that any variant found was causative. And we also tended to only sequence one member of a family. And we now know that there's a lot of what appears to be benign variation in Factor VIII and Factor IX and other variation in that we can't have such a directed approach. In terms of interpretation of variants, the American College of Medical Genetics and Genomics have come up with a methodology for classifying variants as pathogenic and benign and areas in between that. And it's important that we're very strict in terms of our interpretation, and also as a community, we obtain the data we need to be able to call the variants as well as we can for our patients. So one thing we're doing with this project after we complete all of our sequence variation on this initial part is that we will have a ACMG criteria that we have in the supporting information we have for all of our variants publicly available, so other people can go and contribute to that. And we can, for one specific variant, we can use our information as a community to try to understand it. And here's just an example of what we see. So this is hemophilia B patients and the Factor IX variants detected. And so these are 3 patients who have variants, 3 different variants who all had this one Factor VIII variant, which was previously reported in mild hemophilia A. But we also look at a database in patients reported in patients or normals who don't have bleeding disorders, and we use the exact database. And you can see that this has been reported pretty commonly in the exact database, and so it likely is a benign variant. On contrast, this individual has a severe hemophilia B and a variant that is reported and caused in hemophilia B. But in Factor VIII, we found this other variant that's been reported in mild, severe, and also has been reported as being benign. But it's not a very common, it's not seen in this database, and so it raises questions about that variant and could it be having an effect or not? And I think we need more work in that area. In terms of no variant found, our approach was very good at finding variants in the likely causative variants in the areas that targeted for sequence. Overall, it was less than 2% where we did not find a likely causative variant, and most of those were in mild disease, either in both hemophilia A and hemophilia B. So now I'm going to switch to whole genome sequencing. So what we have got a lot of, and a lot of people have contributed a lot of information on genetic variation mutations in Factor VIII and Factor IX in patients with hemophilia. So what would be the value in whole genome sequencing in this population? So one, we're pretty good at finding causative variants, but we don't find them all. So one of them is that we're likely to find additional causative variants, and I will show you one that we found. The other thing is that in some patients with hemophilia, usually severe hemophilia A or B, have large structural variants that affect other genes. And so we can understand that better. Particularly those patients may have symptoms beyond hemophilia. We can understand genes that modulate Factor VIII or Factor IX activity and are genes that modulate comorbidities, particularly inhibitor formation. So we are fortunate to be part of the US TopMed Program. That stands for Transomics for Precision Medicine. And this is a program of the National Heart, Lung, and Blood Institute of the US National Institutes of Health, broader precision medicine activities. So National Heart, Lung, and Blood Institute, or NHLBI, is one of the many institutes at NIH. And the goal of this program is to collect and couple whole genome sequence and other omics data with a lot of phenotypic and environmental data with a focus on heart, lung, blood, and sleep disorders. And so as of January 2018, they had sequenced greater than 100,000 genomes of patients with these disorders, and the plan to sequence greater than 120,000, which they may be at, but I'm not sure. So we had, to date, we've had over 5,000 of our samples undergo whole genome sequencing through TopMed. We are hopeful to have the other 5,000 or so accepted this fall into the program. These sequences, the sequencing for this program, the NHLBI contracts with premier sequencing sites in the US, and this is kind of in reference to the prior speaker, we have to be sure we do this well. And then all of this sequence goes to one, it's actually at the University of Michigan where they put them all together and they do further analysis and annotation. So we're sure we have a uniform data. And what I will, the other point here is these data, there's a large database called dbGaP, the database of genotypes and phenotypes in the US. And they're also, that is evolving into something called the data commons, because as we get more big data, we have to have a way to have that data accessible for analysis and for sharing. And they're dealing with how do we deal with the big data. But it will be available. And we also, I'll tell you a little bit about the research repository. That is available through, will be available through the NIH, but we will be able to link the individuals to the research repository, all not identified, but we will be able to provide that link for studies. So in phase 2, which was the first phase that MLOF was part of, we had 2,181 subjects, and we have some initial analysis from those first 2,000. Now, when they were selected, we were not finished with our project, and so we selected individuals who, DNA from individuals who we had available, and also with an emphasis toward inhibitors. And that's why there are so few hemophilia B with mild and moderate disease in these samples. And so first, I'm going to share some work that Marcia Wheeler, who's a postdoctoral fellow in Debbie Nickerson's lab, and she has a large sequencing program. And Marcia has been looking at structural variants in our first about 2,000 subjects. And these are some of the methodology that she's using. She used to look at structure. So this is an ongoing challenge, but ongoing opportunity, and there are tools being developed. So when I kind of think of that, I have grandchildren now, and so if you have those magnetic blocks that you have to put together in a certain sequence, and think if you had millions of those blocks, and you had to have programs that can put them together and analyze them in exactly the same sequence. And that if you are comparing them to ones that are already there, if you have one little change, it's not so hard to find that single nucleotide variant. But if you have these large structural things where they're all messed up, it could be a real challenge to try to define that well in this pool of so many sequences. And so I will say that we have, to my knowledge, yet to solve the Factor VIII inversion variant issue. Our first 2,000 were sequenced at the New York Genome Center, and of this 2,000, and they used some of their tools, and they could not really characterize the inversions. And in Debbie's lab, Marcia, these tools in her hands really can't characterize well the inversions, detect them. So whole genome sequence at this point is not the place to go to diagnose inversions in the intron 22 and intron 1 inversions. So we need more work in that area, which I'm sure will come. But sometimes we think, oh, we could just sequence everything and we get all the answers. And it's not quite that simple. So what Marcia did find in looking at copy number variants, so large deletions and duplications, she found 72 subjects, 65 with deletions, and 7 with duplications, and all, as you would expect, almost all had severe disease. In hemophilia B, there were 26 subjects with, all had deletions, and almost all had severe disease. And she found no copy number variants in the other gene in these patients. And this was the distribution of the copy number variant. Suggests maybe there's a little more common in these exons, but it is affecting those exons, but it's pretty widespread, and also in the factor IX gene. And this is just an example of the analysis. So these are CNVs affecting factor VIII, this is shown with none, and then you can see there's this deletion, but there's exon 10 is still there, and then here's a demonstration of a duplication of exons 3 through 5. And then for factor IX, we have no copy number variants, a deletion, and of the exon 7 through the 3' untranslated. This was a patient who had a whole gene deletion and part of MCF2 gene. And then this was a very large deletion of factor IX and 15 additional genes. This was a patient with severe hemophilia A, and no candidate variants were found. And there is a deletion in intron 6 in this subject, which may be a regulatory. It was the DNase-1 hypersensitivity site. So it's very possible that it affects a regulatory element in that intron, which affects expression. And we're trying to characterize that a little bit more in the laboratory right now. So multiple factor VIII and factor IX CNVs impacted neighboring genes. In hemophilia A, in these first 2,100 subjects, 19 subjects had multigenic CNVs that included factor VIII, and the largest was about 1 megabase in factor VIII and 50, affecting factor VIII and 50 other genes. In hemophilia B, the largest was the one I described previously. And this is one impacting factor VIII, and the SMIM9 gene, which is of unknown function, has been reported before, and that is common to be affected with factor VIII. And these were the hemophilia A and hemophilia B patients. The large deletion, and shown in bold, are genes that have been reported to have function if they in a loss of function disease. And you can see that G6PD is in here. You may recognize some other genes. So we did find some deletions that have been reported in hemophilia A. There was a deletion described in Moyamoya disease, another gene that's been described as causing X-linked dominant Parkinson's, another with mental retardation and with autism, and in hemophilia B, there's been some reports of MCF2 as a important in factor IX inhibitor formation, and MCF2 is commonly, though, impacted, so I think that needs further study. The other gene we have initially looked at is von Willebrand factor. As you know, factor VIII is stabilized by von Willebrand factor. So if that stabilization or there is lower von Willebrand factor levels, that can impact factor VIII levels. And so when we looked at the von Willebrand factor gene in these patients with both hemophilia A and B, we found 24 variants that had been associated with VWD, and if we use the stricter definition of von Willebrand disease, 16, and these were the distribution which included all different types of von Willebrand disease. In the MLOF subjects, 21.4% had greater than or equal to one VWD-associated VWF variant. With a stricter definition, it was 7.1%, which raises a lot of questions on how there are a lot of issues issues may not be the right thing, but there's a lot of variation in von Willebrand factor and a lot of variation we don't understand its impact on potential bleeding symptoms. But this really shows there's a lot of variation in VWF that might be impacting the phenotype in hemophilia. So in terms of the distribution of severity, in hemophilia A, the distribution was actually across severities in terms of finding, but there was a higher incidence of VWD variants in hemophilia A where we could not find a factor VIII variant. So in patients with hemophilia A, in our cohort with mild or moderate disease in this first 2,100, 28.6% of these 28 where we could not find a variant had a VWD variant. And that's been reported before, particularly Connie Miller at the CDC in the US reported finding VWF variants in patients with hemophilia A in whom the diagnosis, in whom a causative factor VIII variant was not found. We did find some variants that without a causative factor VIII variant that likely are the cause. We found a type 2N homozygous, a novel predicted 2N homozygous, and then we found some compound heterozygotes, 2N with a likely type 3, and we also found a 2N heterozygous, which may affect factor VIII levels, and a novel predicted type 3. We do in our reports back to the center when we don't find a variant in hemophilia A, recommend that the sites test the patient for von Willebrand disease if they have not. So in summary, copy number variants affecting factor VIII or factor IX were common in our first 2,100 subjects in which we have the whole genome sequencing data. There's marked variation in the CMV size and breakpoint location, and they frequently impact neighboring genes, raising questions of syndromes, some of which may be known, but some are likely unknown. The novel intron 6 deletion may identify a regulatory element, and current tools for evaluating structural variation in whole genome sequence do not detect the intron 22 and intron 1 inversions well. VWF variants are commonly found in patients with hemophilia. We don't know the clinical impact, particularly because of challenges in understanding the impact of variants in VWF gene on von Willebrand disease. But it's likely that there are causative variants and certainly modifiers of factor VIII, particularly factor VIII activity. So most of this information I presented at the last part of my talk is absent in targeted approach and may have clinical implications. So I think as we look to the future, as our prior speakers have suggested, that it's complex, but we're likely to see a much better picture of the genotypic underlying of our patients' disorders and also understand modifiers of their disease. In terms of MLOF, so we have a research repository which has serum, plasma, DNA, and RNA, and it's linked, the subjects are linked to phenotypic data in the Athens data set. And then we will also be able to add an additional link to the TopMed whole genome sequencing data. We had our first review of applications, and we expect to have the next review of applications, which is performed by an external scientific advisory board, hopefully early next year. And our first was open to the US community since they had provided the samples, but the next will be open internationally. And so I'd like to acknowledge all of our providers and staff and patients and families in the US who contributed to that, as well as the many individuals involved in carrying out this project. Thank you.